A criminal investigation typically works backwards. First, you find the crime. Then you reconstruct how it was committed. Then you ask the question that matters most. Who knew and when? In the case of He Jiankui, that last question has never been fully answered. What we do know, from his own data, from the investigators who followed, from the scientists who analyzed his work, is that the experiment he conducted was not only unethical, it was scientifically bad. The science didn't work. The safety hadn't been established. And the children who were created to prove his theory are now living proof of something he never intended. That the road to disaster is often paved with the absolute certainty that you are right. Let's start with what He Jiankui actually set out to do. Because on paper, the idea wasn't completely without logic. The gene he targeted is called CCR5. It encodes a protein on the surface of immune cells, specifically, a kind of doorway that HIV uses to enter and infect those cells. Some people are naturally born with a mutation in CCR5 that disables this doorway. They are known to be highly resistant to HIV infection. This mutation, called CCR5 delta 32, exists in nature. It's the basis for one of the only known cases of HIV being functionally cured, a patient called the Berlin patient. So his logic was, if I use CRISPR to disable CCR5 in embryos, those children will be resistant to HIV. The fathers in his study were HIV positive. He framed this as a medical intervention to protect children from a disease that threatened their families. It sounded almost reasonable. It wasn't. When independent researchers analyzed He Jiankui's data, the data he finally shared at the Hong Kong summit under enormous pressure, they found multiple serious problems. First, the edit didn't work as intended. Neither Lulu nor Nana had the CCR5 delta 32 mutation that exists in nature. Instead, each child had developed novel, entirely untested mutations of varying lengths. Mutations that have never been seen before in any human. Mutations whose long-term effects on health and development are completely unknown. He hadn't replicated a known safe genetic variant. He had created new ones. Second, at least one of the children showed signs of mosaicism. This means that not all the cells in her body received the edit. Some cells were edited, some weren't. This patchwork pattern, edited DNA in some tissues, original DNA in others, is a known risk of CRISPR, especially when used in rapidly dividing embryonic cells. It can cause serious developmental complications, and it means the protection he claimed to have given his subjects may not even be complete. Third, there was evidence of off-target edits. Places in the genome where the CRISPR machinery had cut DNA it wasn't supposed to touch. He didn't fully characterize these. He didn't investigate their consequences. He announced his results to a camera before a single qualified peer reviewer had seen his data. And then there is the deeper biological problem, the one that haunts scientists most. CCR5 is not just an HIV doorway. It plays important roles in immune function, brain development, and response to other infections. People who lack functional CCR5 are known to be more susceptible to certain diseases. West Nile virus, severe influenza. The gene he targeted doesn't exist in isolation. It is woven into a complex biological network that scientists do not yet fully understand. By disabling CCR5 to protect against HIV, he may have opened other vulnerabilities. He may have traded one risk for another. We don't know, and that is exactly the point. The scientific community said, "We don't know enough yet. We need more research." He said, "I'll find out using children." What turned this from reckless science into something more sinister was the deception that surrounded it. He Jiankui did not receive legitimate ethics approval for his experiment. Investigation later revealed he had forged ethics committee documentation in order to recruit participants. The consent forms he gave to the couples in his study were written in complex language that obscured what was actually being proposed, that their children would be the first genetically edited humans in history, carrying permanent alterations with unknown consequences. He told participants it was a vaccine trial. He raised his own private funding specifically to evade government oversight. Investigators concluded he had acted deliberately and deceptively, motivated not by concern for patients, but by personal ambition. The parents trusted him. They were not given the information they needed to truly understand what was happening. Their consent was not informed. It was manufactured. Lulu and Nana were not the only children born from He Jiankhui's experiment. At the Hong Kong summit, he mentioned almost in passing that another mother was pregnant. That pregnancy was confirmed in court documents. A third child, later referred to as Amy, was born months after the twins. She too carries edited DNA. She too cannot consent to what was done. She too is part of an experiment with no ongoing scientific oversight, no formal follow-up protocol, and no published safety data. 3 children, 3 permanent experiments, no accountability. In episode 3, we turn to the question of who else knew, because He Jiankhui did not do this entirely alone. And the investigation into what the scientific establishment knew is a story almost as disturbing as the experiment itself. Visit unbedsidemanners.com for more stories.